Hello and welcome to today's ISNTD Connect webinar. I'm Marianne Comperet and I'm speaking from the International Society for Neglected Tropical Diseases. So ISNTD Connect is our weekly get-together around all things in tropical health, and it's lovely to have you here today as we prepare to mark World Malaria Day 2021 later this week. Now, World Malaria Day is, on the one hand, absolutely an opportunity for celebration. The past 2 decades have seen more than 7 million lives saved and over 1 billion malaria cases prevented. I can't help but zoom in, for example, onto the greater Mekong subregion, where the reported number of malaria cases in the last 2 decades fell by 97% and deaths were reduced by more than 99% to just a mere 15. So these are tremendous achievements, and we'd like to join and echo the World Health Organization statement congratulating the growing number of countries who are approaching or have reached 0 cases of malaria. However, let's remember that malaria has not gone away yet. 230 million annual cases are still reported, and this results in about 500,000 deaths. And as we know, sadly, 2/3 of that burden falls upon children under the age of 5 in sub-Saharan Africa. And although this time superbly averted by a range of partners in this field, we also know that a major and unexpected shock, such as the COVID-19 pandemic, could quite rapidly erase and reverse some of these substantial gains. So, against the momentum of this year's World Malaria Day and the renewed support by WHO announced just yesterday to those countries aiming to halt transmission by 2025, let's today take a closer look at a critical strategy for reducing the burden of this disease and reaching that all-important zero malaria target, and that is prevention. Reaching zero malaria starts with prevention, and over these next 90 minutes to give us a comprehensive overview of the landscape, the innovation, and the partnerships forging the path to zero malaria through prevention, it is our huge pleasure to welcome our 4 panelists today at this ISNTD Connect session. And so I'm sure that you will all join me in a virtual round of applause to welcome our panelists. And first and foremost, I'd like to welcome Dr. Beatrice Greco. Hello, Beatrice.
Hello.
Hi, Beatrice. You are Head of R&D and Access at the Global Health Institute with Merck. So welcome, Beatrice.
Thank you.
We will then hear from Danielle Oppong. Danielle, welcome.
Hi.
Hi there. Danielle, you are Managing Director at LivFul Africa, and for those who do not know LivFul, you are a company placing access really at the heart of innovation in repellents in malaria prevention. Next, I'd like to welcome Professor Isaac Quaye. Welcome, Professor Isaac. Hi, you are the President of the Pan-African Ovale and Vivax Network, PAVON, based at the Regent University in Ghana. Hello. And on cue, no longer in the room with us, but no doubt to rejoin very shortly in the next few seconds, we also have Professor Abraham Anang. It's our huge pleasure to welcome Professor Abraham, Director of the Noguchi Memorial Institute for Medical Research in Ghana. So hopefully Professor Anang will just be rejoining in the next few seconds. There we go. Hi, Professor. I was just introducing you. And so,
yeah, thank you. Thank you very much. I'm back on. I'm having
we had no doubt.
Unstable internet, but I believe that it will still be successful. Thank you very much, and I'm happy to be here.
Yes, thank you for making time for us, and lovely to have you here as well. Just turning now as well to welcoming our attendees. So you are no doubt familiar with this, but please, by all means, say hi to us in the chat. Tell us where you're tuning in from. I'd like to say hello to Gaurav Kishan, tuning in from India, Nishe Konda from DRC, Tek Peng Lee, hello from Malaysia. So really audience, see everyone. So without any further delay, I'm going to hand over to our speakers, and first and foremost to Dr. Beatrice Greco to start and take us on this really complex and at the same time hugely important ecosystem of strategies to tackle zero malaria through prevention. I'll hand over to you, Beatrice, to give us a bit of an overview and introduction on that topic.
First of all, thank you very much, Marianne and ISNTD for having us today. Yes, indeed, on the 25th of April, it will be the World Malaria Day under the theme of Zero Malaria, Draw the Line Against Malaria. And this is very much about celebrating also the malaria strategy. A malaria strategy that is based on a number of pillars, and one of them being, as Marianne, you already said, very much ensuring universal access to malaria prevention, diagnosis, and treatments. And what I would like to do today is really to show some of the work that we have been conducting within the Merck Global Health Institute on more activities that are associated with prevention, because we do feel very much that to contribute to the malaria elimination, there's a need to further investigate actions around prevention, hence also the title of our webinar today, Zero Malaria Starts with Prevention. What I'm going to do next is just to introduce you briefly who we are, and then to present a high-level strategic approach that we have applied within our institute, and talk a little bit more on some aspect of some programs. And of course, as you understood, other programs are going to be detailed by the next speakers. So first, just a few words of who we are. So we are Merck, a worldwide company founded more than 350 years ago, that is delivering science and technologies through its three core businesses: life science, healthcare, and electronics. And the purpose of our company is really to significantly impact human lives, and that through delivering innovative treatments, empowering the researchers through access to various tools, and also deliver and develop innovative technologies based on the electronic businesses. We are known throughout the world as Merck, except in the US and Canada, where we are known as EMD Serono, Millipore Sigma, and EMD Electronics. When it comes to global health activities, we have a department that is dedicated to improving the health of underserved populations in low and middle income countries through access to our science and technology, and very much also through works with various partners, because we feel that partnership is anyway the heart of many of those activities. More specifically, our global health strategic intent is to contribute to a number of sustainable development goals. And here we are depicting a certain number where we feel that indeed we can have a significant impact. And from a therapeutic indication focus, we have three major pillars. One being a neglected tropical disease called schistosomiasis. And here there is a strong commitment from the company to support the community till elimination of schistosomiasis as a public health problem. On the other hand, we have activities to prevent and control a number of non-communicable diseases. And finally, we have a pillar on malaria, and here our intent is really to support the community toward preventing, but also controlling malaria towards elimination. How do we do this within our Global Health Institute? Our key focus is really to strengthen low-middle income country health system resiliency in a sustainable manner, in particular in their capacity to prevent infectious disease transmission and increase their preparedness to react to event volatility. The two topic and the two key disease being for us, of course, schistosomiasis and malaria. So how do we do this? It's by providing sustainable access to technological solutions, by supporting access to capacity through workforces, infrastructure building, supporting adequate policy making. It's by looking at our 2 diseases of interest not as a standalone, but within and including a full range of health challenges to be addressed and to provide a more integrated approach. But it's also to look at digital solutions that may help to predict and be aware of upcoming threats. This, of course, we cannot do it alone. And again, this is about building partnerships and working with experts in the field. So when it comes to how we fight malaria, well, those numbers, unfortunately, you know them already, and they have been already mentioned by Marianne, by Danielle. We are at the point where, of course, tremendous progresses have been made, but there's still a lot to be done. And the way we approach it in our institute is by providing and to address it through an integrated approach that is very much aligned with the global technical strategy for malaria elimination, with a key focus on prevention. That means we are looking at developing treatments from drug discovery and clinical development to come up with new anti-malarials that may address more preferentially prevention. It's about looking at how diagnosis and surveillance can really become pillars of, and pillars as standalone pillars almost, to the malaria strategy elimination, with, of course, a keen interest on, again, fostering and surveillance from prevention perspective. Vector control, and you heard about now Daniel's work on IR3535. This is really looking at developing alternatives against malaria transmission, in particular for outdoor bitings. We know that, as Daniel was mentioning, bed nets and indoor spraying are the key arms for fighting malaria transmission from a vector control perspective, as well as, of course, looking at ways to eliminate larvae. But we know also that a lot of biting is happening outdoors during activities. And people do need to have access to safe solutions that are going also to be ecologically safe. And that's the kind of work that we are conducting there with LivFul and the Noguchi Institute. Then we are looking also at the capacity building. And this is a little bit more on the side, but we feel that treatment, diagnosis, vector controls are key pillars, very much highlighted in the malaria strategy. But capacity building and local capacity building by providing access to essential resources is extremely important also. So this is actually translated today in a series of programs that we are conducting through partnerships within our institute. And here you have depicted a certain number of our partners. We are going to mention a few more later on. And you just heard about the work that we do on the IR3535 with the insect repellent. Here, just to give a little bit more details, we are conducting today with LivFul and the Noguchi Institute a series of testing, very, I would say, controlled testing on cage test, then community-based approach, natural settings, to really assure that we are going to have a solid package to demonstrate to the community also that, well, we may have here a way, another tool to really fight malaria transmission. You are going to hear about the surveillance work through the work that Dr. Isaac Quaye is conducting together with us on the PAVON network, and also the basic work that we are conducting on strengthening a state-of-the-art basic microscopy approach, but a work that needs to be done also of strengthening education of healthcare workers when it comes to microscopy for malaria and beyond malaria, looking at also at other elements of infection. So giving a more comprehensive views on the diagnosis, and this is a work that you're going to hear about from Professor Anang. What I'd like to do now is just to give you just very briefly some elements on the capacity building activities that we conduct, plus then I will zoom on to the more treatment elements. From a capacity building, as I was saying, we feel that to tackle prevention, we need really to strengthen health system also at its basis. And this is a little bit aside from a direct, I would say, control of malaria or elimination strategy of malaria, but addressing the basic access to water and clean water in healthcare center as a means to provide the basis to care for the patients that are coming into healthcare centers, post, etc. In a rural setting, it's extremely important. So that's a little bit on a side project, but for us, we feel that this is the baseline that is very important to tackle. The other program that we conduct in collaboration with the Boston University has to do with counterfeited drugs. We know that counterfeited drugs are leading to death, to increase resistance to a number of treatments that are antimicrobial treatments. And this is a huge problem because from the malaria field, because the two top commodities that are mainly counterfeited are antimalarials and antibiotics, two commodities that are being used for treating our patients. So here, together with Boston University, we've been working for the last couple of years on developing a technology that is mobile, that allows, without any particular training, so very user-friendly, a way of addressing whether the active ingredient is the correct one in the drugs that are available. And not only that, but also to quantify the presence of the amount of active ingredient. So it's really to detect substandard and falsified medicines. And we feel that this is a basic also tool that is going to strengthen the capacity of the healthcare centers and healthcare system to really address the proper need of the patients. And this is also a way of preventing re-emergence of resistance and preventing case of death. So another way of prevention. What I would like now to do is to zoom onto some of the treatment activities that we're conducting. And first, looking at what we do from a drug discovery perspective. So we have had the extreme pleasure to have a collaboration, a long-term collaboration now, with partners of Medicine for Malaria Venture, as well as under the leadership of Professor Kelly Shibale with H3D in Cape Town University. We've built a drug discovery hub for malaria. Here was the intent on developing the next generation of safe and efficacious antimalarials with a key focus on preventions. What are going to be the next preventive drugs or chemoprevention drugs? But also, of course, looking at alternatives for treatments and to tackle cure. Today, we are happy to say that we are already in lead optimization and that we're aiming for an early development phase by Q2 of this year. So very exciting platform, a lot of very interesting outcomes to come in the near future. Doing drug discovery was also showing us that the researchers that are conducting drug screening and drug discovery, you do need to have the right tools, and in particular the right cellular tools in their hands, tools that have a capacity to translate really into a human setting very early on. And in particular, again, in the perspective of looking at tools that are going to support the community, looking at more preventive treatment, it was very important to address the question of developing cell assays that would be allowing us to do screening and profiling onto primary hepatocytes or onto hepatic systems that would be recapitulating very much the human liver. And that's actually the work that we've been conducting with IBET and IMM in Portugal, together with our colleagues from Swiss TPH, where we have been developing recently a flexible 3D cell-based assay that is based on a human hepatocyte creating spheroids, recapitulating many features of a human liver in terms of its architecture, but also polarization, metabolism. They can be put in culture for a long time, and we've been doing drug testing on them. So this is an assay that you're going to hear about a little bit more this year. The project lead from our side is Thomas Spangenberg, and I think he's going to be communicating quite a bit on that. And I would advise and recommend that if you're interested in getting access to these type of assays, to get to our colleagues. So this is more from the drug discovery and early phase. Now, if we look more at the clinical development perspective, within our portfolio, we have a compound that is called M5717. This is actually a drug that was identified, a compound that was identified by the University of Dundee together with the Medicine for Malaria Venture team. This is a very interesting molecule because it has a new mode of action. It's an inhibitory of the Plasmodium elongation factor 2. But what needs to be remembered about this molecule is that it has an extremely efficacious aspect or efficacy at all the different malaria stage, liver, blood, gametocyte, mosquito stage also. So it's really a nanomolar range efficacy, which makes it, together with his very long half-life, a very interesting molecule that can become a game changer because it can address all the various target product profile needed today to fight malaria. So it has the potential to become a single dose cure, which is what this field is really looking at from a treatment perspective. It can also become a weekly therapy for chemo prevention, and this is really the space in which we're really looking at very carefully how can we position this molecule as being the next generation of the chemo-preventive treatment. But it has also the potential to become a transmission blocker. So a very interesting profile. Where are we now today? Phase 1 has happened. We have realized and we have demonstrated that it's well tolerated. It has, and the long half-life has been validated in a human setting. And now we are at the step where we have also conducted 2 types of human challenges. So those human challenge are healthy volunteers that accept to be infected either with human erythrocytes containing a laboratory strain of Plasmodium, or that can be infected with sporozoites. And then they're being treated at different times with a drug to be tested. And when we have done this with M5717, what we have demonstrated is that we could reach full cure as well as full protection with doses that are quite nice. So now we are at the next step, which is to find the right combination partner to enter in phase 2. So an exciting time. And we are really happy to have and to be surrounded by partners that are sharing the same type of interest to move this program further. And this is leading me to my last slide, which is, again, to go back to prevention. We feel that indeed the preventive topic has to be strengthened also within the malaria strategy. And it comes with a set of tools that today, hopefully we are going to show and to explain a little bit more. I zoomed onto the treatment, Daniel onto the IR35, and so vector control. But now I'd like to pass on to my colleagues to talk more about the diagnostic as well as the disease surveillance aspect. And with this, I'd like to thank you for your attention, and back to you, Marianne.
A huge thank you, Beatrice. That was really fantastic. The Global Health Institute at Merck, always very committed to transformative innovations and interventions and strategies. And not only are you you show us this very kind of 360 approach to malaria prevention and treatment, but also at every step of the way, lots and lots of partnerships across continents. So very impressive and really fascinating talk. And speaking of partners, as you just said, this leads very nicely to the next presentation by...
Okay, thanks, Mariana. My name is Daniel Oppong. I'm based in Accra, Ghana, and I'm the MD for LivFul Africa. We've been on a journey to help fight malaria on the continent and elsewhere in the world, and it's been a privilege to be part of this journey and our partnership with Merck. Unfortunately, Beatrice couldn't start, but then I'm going to take the lead, and hopefully she can join us and continue from where I'll leave off. A quick introduction. Just when I was about to start.
Sorry, okay. But please go ahead. No problem.
Okay, that's fine.
Sorry, everyone. We carry on with Daniel.
Yes, let's carry on with Daniel, and I will talk afterwards. No problem. Go ahead, Daniel.
Okay. So a quick introduction to LivFul. LivFul is a life science company started in the US. We have presence on all 5 continents now. And our lead product is an insect repellent that we're trying to get to everybody who needs it, and practically everybody in any part of the world where disease-causing vectors are an issue. And as a result of that, we're trying as much as possible to use our live platform to get it to people who otherwise wouldn't have been able to get access to it. Our live platform actually works in diverse ways, and it goes against the conventional way of distribution, manufacturing, and getting products to people. And as a result of that, we are able to reach a lot more people on the lower end of the pyramid and in the mid than it would normally have been possible. How did LivFul start? LivFul is a global company, but then it started from Nigeria, and I see a lot of Africans on the platform, and we should be proud. Our founder, Hogan Bassey, grew up in Nigeria, and at the age of 10, he mixed a concoction in his mom's bathtub to help prevent him from getting malaria. And he says it better than anybody else can, but if you were around that time, you know that chloroquine and other malaria treatments were not pleasant to take. And as a result of that, he said, okay, I don't have access to anything to stop mosquitoes from biting me. Why don't I create something? And that is where the creation of insect repellent started. Thank God he didn't die out of it, but it wasn't anything scientific. He just mixed up some concoctions, which luckily didn't kill him. And we built on that to a point where we have a product that is quite effective. I'd want us to look at the situation with malaria as at the end of 2019 and the world 2020 malaria report. It's quite obvious that there have been significant improvement in the fight against malaria since the year 2000 all the way to 2019. But still, if you look at 2014-15 coming, it's stagnated. That's not to take into account the significant drop in malaria cases, 32% drop in malaria cases between 2020 and 2019, a 44% drop in malaria deaths, a mortality rate reduced by 67%. 1 billion deaths have been prevented, and this is quite significant. Billion cases, over 1 billion cases have been prevented, and 7.6 million deaths have been prevented. Yes, but let's not lose sight of the fact that in spite of all these significant achievements, the burden is still there, and we still have close to 230 million cases, infections were reported in 2019, and out of that, 409,000 deaths were reported. the greater burden of this is on infants under the age of 5. And we all know what that feels, to lose a young child right at the point where they're about to start life. So what can we do to help make this better? Again, this is a quote from Dr. Moiti. She's the regional director for WHO Africa region. And it's a tacit acknowledgement of the fact that there's been significant improvement, and in spite of that, there's a lot more that needs to be done. And there's a need for us to change the way we've been doing things, or add to the tools that have been used in the past to make sure that the fight against malaria is won. LivFul and Merck have been in collaboration in getting to a repellent that is safe, using Merck's active ingredient, the IR3535. It is by far one of the safest, very, very pleasant, and you compare it to others on the market, and you see that this is a unique product. In the EU, is approved for use in young children. I believe Beatrice will talk more about this, but infants as young as 2 months at a certain concentration can use a repellent that's made out of IR3535. It has strong repellent performance against mosquitoes. It's pleasant. It doesn't have any odor. It's safe to use, and it, as I said earlier, is safe for use in all ages, and especially for infants below the age of 5 who bear the greater burden of malaria cases in the affected regions. LivFul's contribution to this is a unique patented technology, STAYTECT, which practically stops the active ingredient from leaving your skin. We encapsulate it, we stop it from being absorbed into your body and evaporating at a greater rate than we want it to. And as a result of that, we're able to extend the time of protection, and also make sure that your body doesn't absorb unnecessarily the active ingredient. It stays on your skin where it's most needed and where it's supposed to protect you against the biting insects. Now, our technology is quite simple. We using natural products to create a capsule that holds the active ingredient on your skin and stops it from leaving. It basically stays there, and that is where it stops the biting insects. And in this case, the most troublesome one in Africa, where I'm based, and also the one that really caused a lot of problem with malaria, the mosquito vector. A complete insect repellent brings together the IR3535 active ingredient from Merck and our STAYTECT technology, and is a groundbreaking innovation in that it's able to take this very potent repellent at IR3535, keeps it on your skin for 13 to 14 hours. It's been tested, and we are very comfortable about its efficacy. Now, a lot of people would ask, why insect repellent and why a complete insect repellent? Yes, we acknowledge the fact that there are a lot of tools out there already. There's the indoor residual spray, there's the treated bird nets, there are other drugs and vaccines that are being tested. Ghana is a place that vaccines are being tested at the moment. But all these tools have their limitations, and as a result of that, we bring in this repellent to complement what they're doing. Insect repellent is something that can be worn the whole day. You can go on a date wearing an insect repellent. It doesn't smell awful, and your girlfriend wouldn't leave you because of it. As a result of that, we are confident that people will use it compared to other products that are on the market that you put on and you smell like you just had an acid or a drug bath. So we are very confident about the impact that this repellent can make in the fight against malaria and other insect-borne diseases, because we are confident people will use it. It doesn't smell awful. It's not greasy. It's practically invisible once you apply it. And as I showed in one of the slides, the active ingredient itself is odorless. Our technology doesn't add any odor, but just to give you a feel that you have a product on and to give you the confidence that you have something, we have a very light scent to the repellent, which is quite pleasant. And we've actually had studies where people say it felt and smelled like body lotion, and they were very comfortable using it. This is efficacy performance of repellent at a concentration of 20% IR 3535. It's very effective, and as I indicated earlier, it protects you from insect mosquito bites for 14 hours. And this is not a test done on just an open environment, but actually in a cage test where we have 200 mosquitoes in a small box, and participants were putting their hand in, one hand protected and the other were not protected. And for 14 hours, we were able to show that the product was quite effective at preventing the mosquitoes from landing. We did a pilot in Ghana. The product was developed in the US. We said, okay, let's bring it to Africa. Let's test it against the Anopheles mosquitoes here. And one popular one that was mentioned to me at Noguchi, the Anopheles gambiae, quite strong breed there. And we said, okay, let's test it. We all know that in our part of the world, malaria is a big problem. And there's a segment of the population that are very much exposed to insect bites. And one of them is the security guards who work in the night. So we partnered with a security company in Kumasi, somewhere in the middle part of Ghana. We gave them protection, 100 security guards, we gave them protection for 45 days. And yes, 97% of them reported insect bites on regular basis when they work in the night. And as a result of that, they had to dress in the night in the warm weather of Ghana. They had to wear gloves, wear long sleeves, thick jackets to prevent them from getting bitten. And even in that case, parts of their bodies that were exposed were still bitten. At the end of the 45-day trial, what we're able to pick from them is that there were no malaria cases reported. Hitherto, the prevalence rate for malaria was about 73% among them, and 99% of them were satisfied with the repellent because it felt good. One comment was, it's nothing like what we've used before. It's pleasant and it's more comfortable to wear it. So that's a key comment, and it's something that's been highlighted as a barrier to adoption for repellent as an effective tool against malaria, because people don't like wearing it when it smells bad. And we've been able to overcome that hurdle with this unique formulation. We said, okay, will you buy our repellent? 94% of them said, yes, we'll buy it. Okay. The company too said, no, we won't let them buy it. We're going to pay for it, because they had direct feedback from premises where they were guarding that your guards are more effective now. We see them making their night rounds. They are not sleeping in corners. They don't lock themselves up in the security cages. You actually see them moving around, making sure that the premises are protected. So the company said, okay, this is adding to the productivity of these people. And as a result of that, we are more than happy to procure it for them. And even at that point, the guy said, okay, we don't earn much, but we know there are people elsewhere in Ghana, specifically up north, who are struggling to get over the burden of malaria. And as a result of that, they were going to donate a percentage of their salary to help us donate some product up north. Then 94% of them again said yes, they will use the repellent on regular basis. They'll buy it, and if it's made available to them. Thank you very much. This is a quick presentation, and I don't know, will we take questions now or?
Hi, Daniel. Well, gave us an overview on malaria, setting the scene, as well as really diving straight into the subject of how to tackle prevention with these hugely encouraging results. So that's wonderful. You did mention that your innovation and your product, it really builds upon a partnership, and I think that makes for a really good segue to the next presentation by Professor Isaac Quaye. Professor Isaac, again, welcome, and we look forward to hearing more about the scope of work at PAVON. So over to you.
Thank you. Great. So I'm happy to present with you the work that PAVON has been doing for the past couple of years in partnership with Merck Global Health Institute. And within the next couple of minutes, I would be expounding on the importance of Plasmodium Vivax as an emerging problem in sub-Saharan Africa, and the fact that if we are not paying attention to this emergence of PV malaria, there's going to be an explosion in the near future, but we hope that will not happen. All right, so we would look at briefly what is the nature of the temporal malaria burden in Africa. I think that if we talk to any national malaria control program and we want to look at what they've been doing with regards to the malaria parasites, they will immediately tell you it's all about P. falciparum. There's very little information on the other parasites, P. malariae, P. ovale, and particularly P. vivax. And so there is some level of neglect of P. vivax at this point. And this is quite unfortunate because it is the most prevalent Plasmodium species throughout the world. And so I guess it's all about the way it has been presented as being benign, and of course the issue of the Duffy antigen negativity that is prevalent within Africa. So I think there are some perspectives that need to change, but I hope that this will be highlighted as we go along. So how endemic is Plasmodium vivax in Africa? Now, this map shows us some data that is available on active surveys that have been done in different countries on the continent, as well as data that has been collected for travelers outside of the continent to either Europe or Asia or North or South America, and then became sick. And then later on, it was found that these individuals had P. vivax and or P. ovale. And we can see clearly that, you know, the red, the circles are for P. vivax and the triangles are for P. ovale. And the red circles are direct surveys that have been reported in the past. And clearly, I mean, except from the northern part, for direct surveys, there's been reported in nearly every country, you know, on the continent that P. vivax exists on the continent, in nearly every country. And then if you combine that with the travelers, you know, who have fallen sick and they've been diagnosed as having P. vivax, then it's entirely covering the whole of Africa that these parasites exist. So the obvious question is that why are we not seeing it, and why are we neglecting it and not paying attention to it? If the aim of the National Malaria Control Program is for elimination, which is really right so, for RBM strategy on elimination and eventual eradication. So there's a perspective on the prevalence of Vivax and PO, which has been neglected. And I think this has to change, and we will show you pretty soon on this. Now, going back to what has been highlighted previously by our previous speakers, Beatrice and Daniel, we are looking at the human hosts with respect to diagnostics, and then the vectors on prevention, and then the environments that allow the parasites to flourish, and then for the vectors to flourish, and how we can intervene on these areas for improved diagnostics, capacity building, and to make sure that we really prevent the parasite from exploding on the continent. So if we look at the human hosts, now the previous thought is that, which is being challenged at this point, is that because in sub-Saharan Africa you have quite a number of the hosts having Duffy antigen negativity, and which is known to prevent or to inhibit the entrance of Vivax into reticulocytes. So then the issue is that you do not expect that in Africa you have any significant number of parasites on the African continent. But the recent evidence is showing that individuals who are Duffy negative are getting infection. This has been reported in several countries, including the surveys that we have done in both Botswana and Namibia. And then, of course, there are vectors that are around, and there are new vectors that come up, with changes in seasonality and changes in geospatial climate profile and the environments. There's always a shifting trend in the transmission profile. And we need to catch up with some of these things to be able to know how to intervene and how to curtail any potential danger associated with the transmission. So I will just bring us back to look at this picture of Duffy antigen negativity in sub-Saharan Africa that I think has been a key issue regarding how much attention has been paid, to Vivax spread in the African continent. And as you can see here, the pink region is the kind of prevalence for Duffy antigen negativity. And so you can actually see that quite a large population within the sub-Saharan Africa are Duffy negative, and therefore one wouldn't expect that they be infected. But then if you look at this, you would have symptomatic cases of infection that are seen here within some of the countries within the regions, and then asymptomatic cases, those who are Duffy positive, which are there, and then the asymptomatic cases are also present, these yellow markings that you find here. So there are areas where you are having an infection because there is Duffy antigen positivity, but you also have in places where you have an infection when they are Duffy negative, and not just asymptomatic infections, but clinical cases of infection are being seen. So the parasite is obviously on the continent and is obviously infecting people, but then we are not paying attention to it. We are just there and not making use of data that is now available. And this is partly because I think the NMCP strategy follows directly from the RBM strategy, which focuses primarily on P. falciparum. I think that kind of image must change, where we have RBM recognizing that Vivax is important on the African continent, and then permitting the NMCPs to now also reassess that indeed this is a problem and we need to partner and ensure that we pay attention to this emerging parasite. And at this point, obviously partnership becomes a key. And I'm glad that PAVON has really taken the step forward on this, partnering with not only NMCPs and the Global Health Institute, but partnering with African Centers of Excellence to ensure that we move this agenda forward to raise the awareness on Vivax. Now, there's an interesting aspect of the biological characteristics of Vivax that makes it particularly difficult, to be able to eliminate. And one of the things is that the normal parasites, for Plasmodium species, human infecting Plasmodium species will normally infect erythrocytes, which are matured red blood cells. But the Vivax infects reticulocytes. These are not so much in number in the blood. And so, and that is one of the reasons why the density of P. vivax infection doesn't go too high. So they choose to infect the early forming red blood cells, and then they can be transmitted as very quickly before the blood cells mature. And then they also have hypnozoids that live within the liver, bone marrow, and the spleen. And obviously, unless you have a drug that is effective in clearing these sites of infection, you normally not do, examine individuals going to take biopsies from the liver or bone marrow to assess infection. So that becomes very, very difficult, to be able to handle this infection. And I think that the new drugs that are coming that are targeting hypnozoids, apart from what we already know, primaquine and tafenoquine, are really welcoming. And I think we are very glad that Merck has taken a lead in this direction. And obviously for primaquine and tafenoquine, we also know that there's a limitation associated with the presence of glucose-6-phosphate dehydrogenase deficiency. And there's not much data on that within the African continent. And so, making use of the available drugs now becomes an issue. One has to also have alongside data on the G6PD deficiency, where individuals who are deficient hemolyze, the red blood cells break very quickly, and that can lead to anemia and more severe disease. So having alternative forms of drugs that is capable of assessing these sites within the human body to clear any hypnozoid infections is absolutely critical in this agenda. And then another point is that in the life cycle of P. vivax, the form of the parasite that is picked by mosquitoes to start the sexual stages is the gametocytes. So the life cycle that starts the sexual reproductive form in the vector, you have the gametocytes emerging very early when there's an infection in the blood. In the first 2 days to 3 days, this would have matured already. And if there's a mosquito bite, this would have been picked, and the transmission would have gone on before the individual can present to a clinic, to be treated if there was a symptomatic infection. If there was no asymptomatic infection, then transmission goes on silently. And so that is another major problem associated with looking at P. vivax and trying to get it eliminated. And then the parasite itself, and then the vectors that transmit it can adapt. The parasite can adapt to temperate condition, tropical environments. It's fine. It will survive. And so it is always available to be picked by an appropriate vector, to sustain the transmission. So we have all these variables that impact the transmission profile, and the seasonality of transmission, the metrics of transmission within geospatial and temporal, spacing, if you want to look at that. And these now have to be very finely teased out, for parasite within in all the African countries, for us to be able to have a comprehensive understanding of how this parasite is moving around and how it is spreading on the continent. And all of these are lacking at this point, except for a few reports here that are coming in from a number of African countries. So we have a real need at getting not only the NMCPs, but also the RBM to refocus our attention on the PV. And we will present to you a case study that we've done, particularly in Botswana, that highlights some of these trends. So from what I've just processed, you will find that once P virus gets into an area or establishes itself, it's pretty difficult to eliminate it. One has to use a lot of interventional activities, understand the transmission process very well, understand the metrics of transmission to be able to target them and eliminate them. And this can change depending on the season, depending on the locality, etc., etc. So our case study was in Botswana, and we did this actively. And as we all know, the NMCP data collection is also based on passive, activity where individuals present at the clinics, and then when they have presented, then the data is assessed on that basis. There's hardly any active case detection that goes on because of funding limitations. So we did this active case detection in Botswana, and this started in 2012. And as you see, at the time, Botswana was essentially at a point of elimination, and it was clear from the map on the left side that there was hardly any PF reported at that time in Botswana. And Botswana was ticked by IBM and WHO that they were going to actively eliminate malaria. And this was very exciting because the data showed that, except that we found that asymptomatically there were hidden spots of PV infection within the country. This was the first report ever. So we obviously drew the attention of the National Malaria Control Program, and but as usual, this is something that is new. It takes time to be able to accept it and then integrate it into the program activities. So we followed up to see whether what we saw in 2012 has changed or what has been the dynamics and the bionomics of the parasites. And so when we did this again, and these were all in school children, and children were largely asymptomatic. There are only a few that we picked from the clinics. And as you can see, in 2016, 2017, we saw that PF had come back very strongly. And not only that, the PV's distribution hadn't abated, actually. There were some reduced locations where in Tutume, where PF came back in Ngami, it was Okavango, Chobe. We didn't do previously, but then we found that they were all hidden in there. And then so none of the PV areas had actually disappeared. The numbers had fallen, and PF had come in, but then they hadn't disappeared. And somehow PF had just come back again suddenly. We don't understand the dynamics, why this happened, what happened. But then we looked at the epidemiological data that was accrued within this period at the time. And that was pretty interesting. And you would find that in 2017, when by 2012, virtually no malaria cases were being reported except for the asymptomatics that we found by molecular assays. 2017, it was all over the place. Malaria was reported nearly everywhere in Botswana. And so at this point, when WHO came in to assess the elimination agenda, it was clear that this was off course. And so there was that recommendation for the country to retool and reassess what had gone wrong and then what had been the problem, and which is being done. And that retooling is ongoing as we speak at this point. And then again, the same profile was seen in 2018. And you can see that, there hasn't been any abatement of, what had happened the previous year. And as we talk, in fact, in the Ngami area, malaria report is already going up. So we see that what is interesting is that what we saw as in asymptomatics actually predicted what was going to happen, what happened epidemiologically as a clinical outbreak. And the reports at the time, was that these were attributed to Plasmodium falciparum, but obviously this will not be absolutely the case. the NMCP had the knowledge, they had the thought that, of course, the focus of the program had been on PF. But from what we have seen and the data we have presented, is obviously not only PF. There was some contribution from PVIVAC, which was unknown because we had done this, in asymptomatic population, but then the outbreak had been seen as a clinical case and had been all attributed to PF infection. And then what we also have seen here is that PV is hard to eliminate once entrenched. The ones that we saw before were still there. They hadn't disappeared, and they definitely were being transmitted. And so the question is, what happened? What's happened, and why did the PF population suddenly come up again? And what was the dynamics that was ongoing? Now, obviously, the first question that we asked ourselves is that, could PV aid in the spread of PF? Now, I've looked at the literature very carefully, and I haven't seen any report on that. It's something that I think one has to begin to examine and think about. Is this something that is happening? Is there a coupling of the infection between the parasite species that is unknown? And so there's some dynamics that needs to be investigated. And then number two is that, how are these trends evolving in other African countries that have now reported on PV? We are seeing many countries in Africa reporting PV presence, not just because they have Duffy positive like in Eastern Africa, but because they are also seeing it in Duffy negative individuals. And so we are seeing that now the parasite is able to invade reticulocytes without as much inhibition as we had previously thought. Is it a new way of invasion, or we don't know? So these dynamics again needs to be assessed. So what is the point here? That the presence of PV and the transmission in Duffy negatives in Africa is a menace. It's obviously a menace. And it's obviously also a ticking time bomb. It is spreading in the asymptomatic population. We don't see it because we are not looking at it. We don't pay attention because somehow we think it doesn't contribute to transmission. And these are areas that I think is well overdue for attention to be given by the NMCPs. And I'm glad that PAVON is leading the way and providing the relevant data that will allow the malaria space and the malaria community, both researchers and management, to be able to think very carefully about how this menace that is coming up can be nipped in the bud. And we would like to thank Merck for the foresight in getting PAVON to get involved in this. Thank you very much. So we will just conclude by this, the way forward for Africa. There should be an urgent inclusion of PV in NMCPs and malaria researchers' malaria agenda, and plans for the elimination is required. We must invest in the active surveys on parasite prevalence, vectors, and their bionomics necessarily. And we have to devise innovative strategies to hinder the spread and potential explosion of the parasites on the African continent. Thank you.
And a huge thank you to you, Professor Isaac, for firmly putting this issue on the agenda. We're really excited to see where the PAVON partnership will be going. It's 2 years old now, and thank you so much for showing us the way so far. you actually started your malaria journey, shall I say, at the Noguchi Memorial Institute for Medical Research 10 years ago now. And so I guess we've gone full circle now, as we will be hearing from Professor Anang, Professor Abraham Anang, who is currently director at the Noguchi Memorial Institute. So for our last presentation of the day, while technology is on our side, Professor Anang, I'm going to hand straight over to you for your presentation. I'll just start.
Thank you very much, Laura.
I'll just start your slides. Thank you.
Thank you very much. I'm very happy to be part of this very important webinar, which is also part of the World Malaria Day celebration and promoted by Merck and others. So my presentation is on healthcare system strengthening through capacity building. And this is really part of a microscopy capacity building in collaboration with Merck. So the outline of the presentation is simple. First, I'll just briefly show what is Noguchi Memorial Institute for Medical Research, and then give a brief on the Ghana malaria strategy to fight malaria and the challenges that are there, then the need for continuous capacity building of health workforce, and then show also the capacity building programs from past collaborations for Noguchi, and then the collaboration of Merck and Noguchi for this particular effort, and then what we hope to achieve. So really, the Noguchi Memorial Institute for Medical Research is one of the world leading biomedical research institutes. The institute is just 41 years old, established in 1979. It was built and donated to the government and people of Ghana by the government and people of Japan at that time. And the mandate of Noguchi is threefold. First, to conduct research into infectious and non-infectious diseases of public health importance. And we know that public health importance in today's global village, a disease in Far East, name it, which China, acute respiratory syndrome, is as of importance in Ghana as anywhere. And the second mandate is to build capacity, especially training of the next generation of scientists, and also to build a capacity of health professionals. And our third mandate is to provide specialized diagnostics and monitoring services, surveillance, in support of public health. So Noguchi was recently just upgraded last year by the addition of an advanced research laboratory, which you can see in this slide. And this laboratory is bringing into sharp focus the works done on virology, bacteriology, and also in immunology. The Ghana malaria control strategy is very well established, and Ghana really is an endemic country. Malaria is perennial in Ghana. It has been with us for ages, for centuries, and the population 100% is at risk of malaria. And malaria incidence in the country is reasonably about 270 plus per 1,000 population. And really the impact of malaria is high because about 40% of outpatients department visits to hospitals are caused by malaria. The strategy of the National Malaria Control Program of Ghana really is based on an integrated approach of building the capacity for control and elimination and eventually eradication of the disease. But it's looking at reducing malaria morbidity and mortality to about 75% of what used to be the burden in 2012, and to reach a target of 75% reduction by 2020. The key inputs into the strategy are overall health sector development, improved strategic investments in malaria control, and increased coverage, towards universal access. But really, there are three major thrusts. That is based on prevention of malaria through, let's say, intermittent preventive treatments, IPTP, for pregnant women, for infants, insecticide-treated net usages, and IRS, and also the second thrust is diagnosis and treatment, and this also focuses a great deal of effort for treatment, for diagnosis, for example, on the use of microscopy for diagnosing malaria, and also the use of rapid diagnostic tests. And the third thrust is basically mainly based on surveillance. So surveillance, including DHMIS, including data quality, issues, and also research into households and science-based, action. Basically, the situation of malaria in Ghana appears to be increased cases, but really this is not because there are increased cases. The efficiency of control of malaria and management in the country has improved. So increased cases are due more to the improved access to healthcare by the populations, the laboratory testing which is enhanced, and also the surveillance. So a lot has been achieved since this program has been ongoing, but there are still quite something to be done. Basically, malaria microscopy gaps and challenges are there. Malaria microscopy, is the gold standard for diagnosis of malaria. So really, globally, microscopy is being used to do a lot of, the improving the control. However, much of the microscopy is being done by very few countries. For example, India is doing most of the millions of microscopy done. Let's say 2017, about 208 million patients were tested by microscope, but mostly in India. In Africa, there are far less testings being done, and this is mostly also due to fewer quality assurance programs and quality or expert microscopists being available to carry out the task. Microscopy basically is a useful tool, and preparing samples for blood samples for microscopy is not so, and it takes some time, but not so much. Like an hour you can finish preparing a slide, and an expert microscopist can read a slide in about 20 minutes, which is quite encouraging. And the level of detection of malaria parasites in slides that have been prepared is about 10 to 50 infected red blood cells per microliter of blood. The cost of microscopy is not too high per se, about $4 to $5, dollars, so $0.4 dollars per slide, plus technician time. So basically, the World Health Organization has come up clearly with the health system framework, which has 6 thrusts, including health workforce. And the health workforce is a very, very important part of the health systems framework, because it has direct influence on access and coverage, and also quality and safety. And with the trained workforce that is up to the task, we are able to also leverage improved health, leverage responsiveness, social and financial risk protection effects are achieved, and there is also improved efficiency. So building the capacity and competencies of the workforce is really a critical aspect of the malaria control effort in nations and globally. At Noguchi, we have built, we have a history of capacity building, organizing various training programs that have been done over the years to strengthen malaria control and others. One of them, which is very important, is a training program that was promoted and sponsored by Japan through JICA, the post-Ebola laboratory capacity building third country. And in this program, the Noguchi Memorial Institute for Medical Research is the implementing institution that provides the training. Japan provides the sponsorship. And the third countries, this training has trained technicians and build capacity for laboratory, technologies from 11 West African countries so far. And we also run various other training programs, including one very interesting one, which we have been running with the US African Malaria Task Force, that has been building the capacity of technologists who are working in military health facilities. And then, of course, we have other training programs for building capacity in bioinformatics and genomics and things like that. So basically, our partnership with Merck, we are looking at a partnership that would contribute to building health systems capacity in Ghana. We are looking at this partnership that will enhance greatly the laboratory technicians' capabilities for microscopy. We are looking at a dedicated program that we call it GALAC, which means Ghana Laboratory Capacity Building Program. And we are focusing this training program on an objective to build accuracy in diagnosis of the disease. We also are looking at a program that will naturally be able to train the technologists in a way that they can take ownership of the programs that are involved. The partners that are involved in this program are mainly 5. The Noguchi Memorial Institute for Medical Research, involved in selection of candidates, supervision of candidates, post-training and continuous education. We have the Ghana Health Service, which is doing also main selection and then supervision to ensure that the candidates participants are fairly and adequately distributed across the country. And Merck, which is the main promoter of this program, is providing organizational and facilitating the training materials and sponsorship of the various modules that are required. We also have the Rotarian Malarian partners who are providing funding for malaria stations and continuous relationship with the Ghana business community to ensure good degree of sustainability. And then we have the foundation partner, which is enhancing communication, advocacy, and stakeholder, and to reach the community and link to the health services. What we hope to achieve together in this effort is to strengthen the confidence of the workforce, improve their skills, and of course also provide a knowledge-based, evidence-based capability in the workforce ability to deliver. We also are looking at fostering a sense of ownership and empowering the trainees to be able to deliver on a long-term basis. And we're looking at having training that will enhance the ability of the trainers to deliver quality management systems and also build international standards, ISO accreditation in laboratories for malaria diagnosis. The long-term success or sustainability is a key issue in this effort. So I'm happy to communicate this, and I would say thank you, and we expect to see good smiles and a bright future, and of course a successful control of malaria for the future. Thank you very much.
Thank you so much, Professor Abraham, for this. We got from the gist of that presentation absolutely is the huge commitment to capacity building, as well as those very practical steps that you've explained on how this will be a major component of prevention in malaria. So a very big thank you for that. And again, that concludes our presentations for today. So a big thank you to the 4 speakers who, in this very short amount of time, actually, you've managed to give us a really important and comprehensive overview of really the agenda moving forward in terms of malaria prevention and malaria control, with hugely important lessons as well, and partnership models for anyone working more acutely in an NTD. So thank you, all of you, for that. And if you're familiar with ISNTD Connect, Cameron, my colleague, and I, depending on who's hosting, normally might have to really kind of encourage everyone to ask a lot of questions or be active in the chat, but clearly today this has absolutely not been a problem. Every single one of those presentations has actually generated a deluge of questions for our panelists, so lots to explore there. We have unbelievably already come to our 90 minutes, and as I was going through the chat, I noticed quite a few of the questions are fairly specific or rather technical, and so perhaps with your permission, panelists, we could send you individually those very specific questions, although again, I see many of them have actually been answered in the chat itself. Perhaps just by way of concluding this session, since we probably won't have time to go through all of those fairly acute questions, perhaps I could all invite you to, well, rejoin us, perhaps with the cameras on, and ask all the panelists just one question, which is where this webinar started, which was all about achieving zero malaria, but through prevention. And so I wanted to ask you both as a more general way, but also as a way of bringing all your approaches back together and concluding this session, for in your field of work, very specifically, and you know, from your experience, at this point in the journey to zero malaria, why is prevention so important? Perhaps Beatrice, you might want to go first. Maybe you don't want to.
Well, thank you.
I'm reverting back to the planned order, shall I say?
No, but I think it's quite clear that a lot is happening when it comes to, I would say, treatment, to address case management, etc. But we see with the science and the type of work that, for example, Isaac is doing, that there's the need to really go beyond treatment and case management. There's the need to really push the strategy of prevention in a larger sense. And that's what we were trying to show. So it's in the sense of, okay, it's about vector control, and here there's a lot to be done. But it's also about putting the tools, such as the surveillance tools, in place to prevent further outbreaks, or to at least predict, if we can reach to that point of prediction of outbreaks, then together with indeed the strengthening of those health systems to have the right tools to address this immediately before it becomes really an outbreak. So I feel that we are at a time where the field of malaria has matured to the point where prevention is becoming a topic by itself that needs probably a little bit more thinking. And that's probably where I will stop.
And that's a great place to stop, because I suppose just following through the running order of the presentations, I was now going to turn to Daniel. And one thing you really showed very nicely, and you were emphasizing this, was that repellents would come in as a complementary strategy to lots of others there. And we don't need to ask you why you feel prevention is important, but do you find that perhaps something like repellents has lagged in terms of advocacy and communication? So, of course, prevention is hugely important in your field of work, but also really does this need to be shared? The advocacy for specifically repellents need to be stepped up?
Certainly, certainly, because if you look at what a lot of the spending that goes into malaria prevention goes into, repellents are not that much on the radar, and that's what we're working up against, and that's what we're trying to correct, because repellents are shown to be they are beginning to show to be quite effective against malaria and other vector-borne diseases, and it's our mission to make our repellent available because it's unique, it actually works, and it's not it doesn't have all the negatives that other repellents have shown to exhibit, and which as a result have prevented people from using it even when they get access to it. And our unique formulation really addresses most of the challenges and the negatives that have been associated with repellents for so long. And we believe with a lot more access to our repellents will become very prominent in the fight against malaria.
And do you feel that perhaps maybe repellents have been repellents' worst enemy, in the sense that some repellents that perhaps weren't efficacious or have changed public perception of repellents? And do you think this will be changing going forward?
Yes, certainly, because let me use us as an example. We see repellents on the African market that have lower concentration of active ingredient because of pricing and everything, but our repellent has the same quality everywhere you go, even though our model allows us to do packaging locally in countries where we operate. The formula is the same everywhere you meet the repellent, and as a result of that, you can get the same quality you get in the US, in Europe, anywhere, in a village in Ghana or in a village somewhere in Chad or wherever you find yourself. We believe that's the only way we can address the bad perception about repellents, making it available and also maintaining the quality across board.
Mm-hmm. Fantastic. And I believe your colleague, Bettina Maxam, has also been posting links and resources in the chat. So if anybody would like to find out more, please go over to the chat. I'll move on now to yourself, Professor Isaac. Just a kind of concluding thoughts. Why is prevention so important? Although, of course, we got a really good flavor of this from your presentation, highlighting all those huge gaps.
Yeah, exactly. I mean, there's a common adage that says that prevention is better than cure. And I think once you get an established infection, the dynamics of treatment, it becomes more complicated. And so it's better to prevent that complication than allow it to settle and then deal with all the different things that comes up. So I think starting to ensure that we don't allow, for instance, the parasite to take hold, before now we begin to look at all the treatment, diagnostics, skills, training, and etc. So we have to put in a lot now to ensure that it doesn't take a hold.
A very striking and concrete example there supporting that. So thank you so much for your presentation and your ongoing work as well. We look forward to following all of that. Thank you, Professor Isaac. And finally, Professor Abraham, your final thoughts on this? Oh, Professor Anang, can you hear me okay?
Yeah.
You were on mute.
I can hear you.
So, same question to yourself, Professor.
Beautiful comments they have made. Yes, the answers they have given. And I would also want to add that, as Isaac said, prevention obviously will reduce complicated malaria, which is a very good thing for all of us, especially in the endemic countries. And then again, prevention will also reduce the workload on the workforce and free extra hands to address other health situations and emergencies. And then the other important thing is that prevention will do one great thing for us, because as we allow disease pathogens to cycle through the population, we also give them the opportunity to go through the various evolutions, mutations, and come up with emerging and re-emerging disease threats. So prevention would do us that great favor. And finally, I would say that by preventing, a lot of gross national, GDP that is lost in developing countries is going to be recovered. So surely I agree with Beatrice for highlighting prevention in this situation. Thank you very much.
Yeah.
Thank you very much. You've left us with much food for thought. For anyone who would like to find out more, please don't forget to consult the PAVON website, the Noguchi Institute's website as well, following all this work. And of course, again, some more resources and links posted by Claudia Secalupo from Merck about all the numerous, varied and incredible partnerships and pretty cutting-edge projects being spearheaded by Merck. So lots to follow up on there. I have to say, you've made my World Malaria Day 2021. I'm sure the audience will join me in thanking you and just reiterating how much we have learned today from all of our speakers and presenters. So a really massive thank you. I'm very sorry we couldn't get down to all the questions and the really important things that were being picked up on by the audience. But perhaps what we need now is an hour and a half with each of you individually, just to drill down into the detail. But until then, I just want to say a huge thank you again, and see you very, very soon.
Thank you very much, Marianne. And thank you also to our colleagues.
Thank you.
Thank you.
Thank you.
Thank you.
Yeah.
Bye.
Bye.
Bye. Take care.
Bye.
Bye.
Thank you.